Good afternoon, everyone, and thank you very much for being here. Special thank to our guest, Professor Jorgensen and Professor Woda, for today's CECAM Marvel Classics in molecular and material modeling. This is a series of events. This is number 9, that together with Marvel, we are organizing to have two kinds of discussion. The normal scientific discussion with eminent representative of many different communities. Today is computational biology and computational drug discovery. And then the second part in which there is an informal interview touching also aspects related to interesting anecdotes of Bill and Shoshana careers in academia and in computational biology and computational drug discovery, in this case in particular. As I said, this is a series of events organized together between CECAM, that you know pretty well, and Marvel, that you know probably as well as CECAM, but for our guests, Marvel is a National Center for Competence in Research. Is a pretty large Swiss National Science Foundation project. The director is Professor Marzari, was started in 2014, and will last until 2026 for about 12 years. And the center is dedicated to computational method for designing and for simulating next generation materials. So as I said, in this lecture series, we take different fundamental developments in simulation and modeling, milestone in many different fields. Bill and Shoshana, they contributed with very, very important discovery and the development of critical methodologies for computational biology, Shoshana, and for computational drug design and discovery, Bill Jorgensen. And as I said, there will be initially a 40 minutes normal scientific seminar, which then will be open to the normal Q&A. Then we have a break of 10 minutes, and we organize the informal interview, where we may ask also general questions to our eminent guest related to science, related to life in science, related to academia, industry, new development, new trend, the AI hype, probably, or the AI breakthrough. And so we may really have a very, very nice and informal conversation with our special guest. These are the upcoming events. I would like to recall everyone, CECAM 55, which is probably the most important event for our community. Our community is a pretty large community, statistical mechanics and electronic structural calculation, to use a historical keyword, but more and more we are moving in the field of machine learning and artificial intelligence. Will be in September between 2 and 4, 2nd and 4th of September, and we will celebrate 55 years of CECAM, which was founded in 1969 in Paris. And then the next CECAM Marvel Classics, which will be in 2025 with David Shepperly and Claudia Filippi, here again in this room, January 27. And without any further delay, I start presenting Professor Jorgensen from Yale. Bill is professor in Yale has been professor in Yale since 1990. And in the field of computational drug discovery, he developed many, many important computational tools, ranging from the OPLS force field, which is now widespread utilized in the field of computational drug design. The TIP model, the TIP water model, TIP3P, TIP4P water model. Again, a very important model for explicit solvent calculations in biology and drug discovery. And many applications of this approach and many other methods and many applications of this approach to real-life drug discovery program. He was able to discover molecules with a very, very strong potency in the field of reverse transcriptase, in the field of anti-AIDS molecules, and more recently in cancer and infectious diseases. Bill received many awards, many prizes. He's member of important international scientific organizations, just to mention the Tetrahedron Prize and the Arthur Cope Award, and his membership of the American National Academy of Science. Today, Bill will report to us about evolution of free energy calculation and historical evolution of this methodology applied originally to the butane, which is a very, very small organic molecule, up to drug discovery programs as he is doing today in his current research activities. Please, Bill, thank you very much again for being here, and the stage is yours.
Thank you very much, Andrea, and it's a pleasure to be here. I thank Marvel and CECAM for making this possible, and I'm happy to see people in attendance in the room and also online. So it's a pleasure to tell you a little bit about the evolution of computer-aided drug discovery, and this is my research group right before COVID, and some buildings at Yale. Our labs are in the newer building there from 2005. We'll see if I can advance. All right, so this is the current sort of scheme of computer-aided drug discovery that we pursue in my laboratory. There's also, I think, a common flow chart for many people's efforts. So there's a lead identification part. First of all, you have to have a target. The targets we've worked on, the target of most drugs are proteins. This is structure-based drug design. So we do need a structure of the protein, which we obtain almost always from crystallography. In our work, very often the crystal structure exists for the protein of interest bound to a small molecule. So what we do is we remove the small molecule and start over to develop our own inhibitors that presumably have some benefits over what was originally in the crystal structure. From there, we do virtual screening, and that can be of two forms: docking, that I think you're familiar with, and then also de novo design. So I wrote a program more than 20 years ago. It evolves, called the BOMB program, that's in the area of programs that are called growing programs. And I'll show you one slide on BOMB, but you start with a kernel, a small molecule in the binding site, and then the program evolves new molecules from that. So it can build huge libraries bound to the target in question. And then these compounds that you either design with BOMB or that you decide from your docking to purchase, you have to then come in possession of them. You have to have an assay in place. And then this normally leads to micromolar hits. So these are compounds that are active in an assay with IC50s typically, or KDs in the micromolar range. To be a drug, though, you have to be much more potent. And this just has to do with human pharmacology and typical doses of drugs. So you really need to be typically in the low nanomolar range in the assay of typical relevance, an inhibition assay or a cell assay. So that's where I'd say the hard work, most challenging work begins, which is lead optimization. So taking the micromolar hit and improving it. And so we improve it by doing a lot of structure building, generating new analogs of the original hit using our bomb program, and also then doing free energy perturbation calculations. So I've committed a large effort over the last 30 years to developing FEP methodology to be truly useful for lead optimization. I think this is now well accepted. Virtually every major pharmaceutical company is doing lots of FEP calculations in the way that we showed was viable more than 20 years ago. So then you take the best compounds from the predictions. You have to synthesize them. You have to assay them. We also try to get new crystal structures of the compounds bound to the protein. And then you repeat this cycle until you get to the desired level of potency, usually in the low nanomolar level. There are issues associated with this. The compounds that you end up with need to be drug-like, meaning that they have reasonable pharmacological properties. And to help in that regard, I wrote a program that's quite popular in the early 2000s called QuickProp. It's distributed by Schrodinger. I'll say a little bit more about it. So it's an AI ML program from the early 2000s, trained on experimental data to make predictions on a wide range of useful pharmacological properties. There's other issues involved in this. Synthesis is really requires great patience. Most reaction sequences that you design on a piece of paper, you know your sophomore organic chemistry, and you can write down the roots. And what you find out in the lab is probably about half of the things that you try in synthesis fail. They either don't give you the product that you want, or it's in terrible yield. So that you have to be very persistent with synthesis. So the people who do it are special individuals. And then the assaying, I won't get into details there, but you have to be very careful if you're a chemist and you're working with collaborators or biologists who are doing the assays to make sure you understand the assays very well and that you can see potential pitfalls. So how did we get to this point where we have this chart and this way of proceeding? And so that I give you some dates here. So now we're going to go back in time and talk about how we got here. And so I won't cover every topic. And this is a relatively personal view, but I have been there since the beginning almost. So first of all, let's see if I can. Okay, so starting at the beginning, I mentioned something. I started my independent career in 1975. So I was just 25 years old at the time, so you can do some easy math there. And I worked prior to that for E.J. Corey, famous synthetic chemist, and I worked half-time for him, and I did chemical physics the other half-time. But the half-time I worked for Corey, I worked on his LASA program. This was a program that did synthetic design. You would draw a molecule on a graphics terminal and work backwards, giving synthetic roots. And this type of programming, this was done in the 1970s. Corey's project continued into the 80s. It was what was noted in his Nobel Prize from 1990. But this is classic AI from the 1970s. And then when I started my independent career in 1975, I continued a project in that vein in addition to our quantum mechanical projects that we were doing at the time. And this was the Cameo program. So this was a program where I wanted just to address the problem. If I know what's in the pot, can I predict what will get out? So if I know reactants and the conditions, can I predict products? And this was done in a very AI manner. So AI has been around in strong form since the 1970s. There was a lot of discussion of AI in the computer science literature in the 1970s. The computer synthesis field was very active in the 1970s. In Corey's program, for example, we described that as a rule-based expert system. So this is, again, classic AI. So there's nothing new about AI. And so I was very involved in that type of programming. I've always been a very active coder going back to the 1970s. I subsequently decided, though I had had a background also in quantum chemistry, that I really wanted to study chemistry in solution because that's where most chemistry occurs, and including, of course, in biological systems. So in the 1970s, this was very early days for doing any type of molecular dynamics or Monte Carlo. By the end of the 1970s, very little had been done in fluid simulations on pure liquids, other than water, Leonard-Jones type fluids. Just very simple things have been done. And part of this was because there wasn't enough computer time. The computer resources were very limited. There also wasn't any software. You see, there was no Amber or Charm. They started to be developed later in that decade, and then very much in the 1980s. So there's no software and there's no computing power, so it's very difficult to do liquid simulations. So I, however, wrote code, got into doing Monte Carlo simulations, and started with simple liquids like HF and then water, and developed at that time what we called the transferable intermolecular potential functions, and started doing some simple questions like studying solvent effects on conformational equilibrium, which is a free energy phenomenon. So that's 1980. Well, this led to, in 1983, development of the water models that most people are familiar with, tip 3P and tip 4P. The advance here was we finally had enough computer power to do iterative Monte Carlo simulations, so we could optimize the parameters so you could get correct properties for liquid water. Also, by that time, we had implemented the NPT ensemble in my Monte Carlo code, which is relatively straightforward comparing to MD. That's why MD very much trailed on this pure liquid simulation. So with my Monte Carlo program, for example, I did the first simulation ever of things as fundamental as liquid methanol. And then, and because you can do NPT, you could compute heat of vaporization, but very importantly, you can compute densities. And so prior to that, if you're doing NVT or NVE, you're just fixing the volume. And what turns out is that you don't get density right unless you can compute it, because it's very sensitive to the Leonard-Jones sigma parameters. So the big advance here was to do iterative simulations, to have the software, and to do the NPT ensemble. So that delivered the water models. Then when did free energy calculations start? Well, that was happening a little bit by 1980. The principal method that was being used up to that point was umbrella sampling, which had only been developed and discovered in 1975 by Vallot. So it wasn't until 1985 that we did this initial ethane to methanol FEP calculation. And why was that? Well, again, now you had to run a series of simulations. When one simulation was painful, to run a series was really painful. Again, no software. So I had to write the code to do this. It was very exploratory. This is Swanzig's paper from 1954 that we now consider to be a classic. In 1984, had 8 citations. Okay? In 1985, it had another 8 citations. So this was not something people were looking at. And then the citations were referring to things like perturbing a a hard sphere to a Leonard Jones particle. Nothing like an organic molecule. So this was very exploratory, but it worked out very well. And then that began other people trying to do these FEP calculations. So Shoshana will also talk about some of this history and even more on focusing on the proteins. But until you had the water molecule, enough computer resources, nobody was doing simulations of proteins in water. The proteins in water didn't happen until about 1985 or so. And even then, it was just a few research groups doing simple studies. And it wasn't common until the late 1980s. So this is, I point out, 1988, there was a meeting in Sweden with a number of research groups that were doing MD on proteins in water. It was just a handful of groups. Berenson's group, there was a group in Lund, my group, and just a couple others, Peter Coleman's group. And so in 1988, we reported 100 picosecond simulation on an ovomucoid. So everybody else is doing BPTI, so we decided to do something a little different. So ovomucoid is another protease inhibitor that's very similar to BPTI. But I worked on this because a person at Purdue across the hall from me is Mr. Ovomucoid. So he had a biochemist who had isolated ovomucoids. So that was state of the art, 1988 or so, doing a 64 residue protein with perhaps 1,000 water molecules for 100 picoseconds. So continuing then to get into protein ligand binding, first you study more simpler systems. So we and others, including people associated with Jean-Marie Lane's group, and also other research groups, and Andy McCammon, worked on some host-guest systems, so organic host-guest systems, very much contemporaneously with the type of work that was going on in the organic host-guest community. And these are some of the systems we looked at, tweezers in the bottom, and various cyclophanes, etc. And so that was the size of systems where we're getting our feet into doing ultimately protein ligand binding, where we're doing again predictions of the free energies of binding. So this continued. We started in 91 here, doing calculations where we were unfolding a little protein, S peptide, trying to get insights on protein folding by time reversal. So this is MD. 1990s also saw the development of the all-atom force fields, the CHARM, AMBER, OPLS. Prior to that, we used, quote, united atom force fields where hydrogens on carbon were implicit. And so it was kind of amusing when coarse graining came along, which was reversing. So the desire was in the 90s to have force fields more accurate by including all of the hydrogens, which does let you do better on torsional energetics. And subsequently, maybe in the 2000s, people want to go backwards and do coarse graining. Of course, the questions you can ask with coarse graining are quite different when you have these all-atom simulations. So this continued. In 97, I modified the Monte Carlo program, and I wrote a separate program called MC Pro that lets one do Monte Carlo on proteins. I think Monte Carlo has been very under-investigated because there's been too much on MD by comparison, but you can do interesting types of importance sampling, if you will, moves with Monte Carlo. So I won't have to speed up here a little bit, but then, coming 2000s, we had enough confidence in our protein ligand binding by FEP that we wanted to design molecules and test them. So we're going to have to now put the predictions to experimental tests. And that really started in the early 2000s. It also caused me to get involved worrying about ADME properties. And this is when I wrote the QuickProp program, which I'll show you just one slide on, which was in a very early AI program for predicting a wide range of pharmacological properties, all trained on experimental data, mostly done with regression equations, some pattern recognition. And then the BOMB program, I wrote to do the de novo design. Then starting in 2004, I began an experimental program where it's synthetic chemists, assay people, crystallographers, all in our group, actually making inhibitors. Our first target was HIV reverse transcriptase. This continued on, and I will show you the latter part of my talk will be about our work on developing SARS-CoV-2 main protease inhibitors, which we did very rapidly due to these methods that we have evolved. Another point here, you might say, well, when did there become more general interest, and when did Schrodinger get involved? And the answer to that is Schrodinger's first FEP calculations, I've been associated with them for a long time. They took my MC Pro program, I gave it to them, and they created MC Pro Plus. That was the first foray of Schrodinger into FEP calculations. And that occurred starting in 2006. It wasn't until 2015 that FEP Plus came along, which is now so heavily used across academia and industry. So the Schrodinger started with my MC Pro program. And then continuing, I will get into work on protease inhibitors. All right, so here's a QuickProp result. Quick means it's fast, so you can run, a molecule takes like a microsecond to run. So here's a molecule fentanyl, and this is the type of thing that QuickProp predicts. It predicts metabolic possibilities, and this is a pattern recognition thing. I wrote every line of QuickProp myself, and the same with BOMB. And this is all because of the need to have this type of information. And then it makes predictions on, for example, log P, so optimal water partition coefficient. What's in parentheses here is the result for known oral drugs, the range. So fentanyl is a log P of 4.26. I have a second way of predicting it by similarity to known compound, where I keep a database, gives very similar results. Solubility is very important, saying what it is, 10 to the -4, serum protein binding, blood-brain barrier penetration. So it says fentanyl is going to get into the brain, which it does. So predicted CNS activity is plus, the highest would be plus. hERG, ACO, human oral absorption. These are all things that are predicted in like I said, a microsecond, because I've trained it on experimental data. It also tells you compounds, fentanyl, the input molecule is similar to. And here are the top two that it predicts, and this is done by a similarity match Yamamoto score. So again, here I am, somebody who started out as a quantum chemist, but I learned through graduate school about AI and ML and dealing with things like Yamamoto coefficients. All right, so the BOMB program is a growing program, and it adds substituents to a core. Here's a core, for example, benzene, or phenol actually, put in the binding site. This is done by the person who's using the program. This happens to be a protein called MIF. And then you have sites where you can substitute, and BOMB has a library of about 800 things that you can put on there. So every conceivable substituent, every conceivable heterocycle, and then it also has different topologies. You can sprout them 4 at a time from the core, or you can link them 1, 2, 3, 4. You can make very long molecules or any combination of those. So this type of program has been around for a while, but this is my personal a flavor of it, very much designed to build drug-like molecules. So here is what, just adding a couple of groups, just two heterocycles I've attached. So a 5-ring and a 6-ring heterocycle, you get molecules that look like this. Then you have to evaluate them with docking type scoring functions that I've trained on experimental data, just like docking, and it has the same sort of limitations of docking. But that's the growing program. So then FEP calculations. So where did that come from? Well, this is just very briefly, this is Gibbs, who's a professor at Yale, Kirkwood, who's also a professor at Yale, and then Perels, who was a physicist, who's, I think, under-recognized in this area. But of course, Gibbs developed delta G, and then Kirkwood had this thermodynamic integration, 1935, which is still used to get free energy changes. Statistical perturbation theory, you can see in Landau and Lifshitz, 1951, it traces back to Perels, 1933. There's an expression involving energy changes to get free energy. And then Zwanzig's paper from 1954 is just a generalization of this, where you can see if you expand this to first order, you get what I call the Perels equation. And so this is called statistical perturbation theory. At some point, it got morphed into the name being FEP. Okay, so we don't exactly know what happened. When I started doing it in 1985, you look at our paper, we call it SPT, statistical perturbation theory. And then Torrey and Below, umbrella sampling is 1975. An early application, so again, by the end of the 1970s, there was very little going on in free energy calculations because of what I said, computer time and software. And so early applications were really things like butane and Leonard Jones liquid by Bruce Burns' group and my group in 79 or 80. And then this is now 1982. The first simulations of butane in water were done by Burns' group and by me in 1982. Okay? And so at this point, we could compute the solvent effect on the Gauche anti-equilibrium, seeing that in water you do get more Gauche, consistent with the hydrophobic effect. But that was state-of-the-art stuff in 1982. Here it's Zwanzig's paper from 54, and then our 1985 paper where we, it was the first molecular FEP calculation where we did the modern FEP calculation where we did the windowing. This is what was now referred to as single topology for FEP calculations that I coded into my Monte Carlo program. The beauty of this was the precision. You could get free energy changes with very little uncertainty, 0.2 of a kcal per mole. And that caught a lot of people's attention. All right, for protein ligand binding, you're probably familiar with the cycle. So you have protein green ligand get complex, protein blue ligand get complex. What we do in the computer is we just mutate the green ligand to the blue ligand. That's just the ethane to methanol FEP calculation. And then you do it for the complex. The difference in the numbers gives you the change in free energy of binding. We do this normally. You have a protein, a ligand, and a couple thousand water molecules in a ball. So if you can answer that question well, then you can just try different changes to your ligand and obtain predictions on how to improve the binding. So the type of thing that was being done circa 2000 was all just retrospective studies by my group and other people's groups that is answering questions like here for a COX-2 inhibitor. If you change the methyl group there to these other groups, how do you compare with experiment? And so we and others have done enough of this to see that you could predict quite well the outcome in comparison with experiment. So we got enough courage from that to really try to do prospective work. So you don't know the answer ahead of time. So you're going to have to design molecules, make them, assay them, and see if you're right. And this required continued evolution, both software, force fields had to improve, computer time so you could get proper convergence. But by 2009, we had enough good results working on HIV reverse transcriptase so we could have published this Accounts of Chemical Research article. So here are some of the early results from 2006, where we're making HIV inhibitors. And I'll just highlight one. So this is a typical thing where you're optimizing substituents or you're optimizing ring systems. So I'll highlight these three compounds. If you can see them well, they're isomers. So one's an indole and then these indolazine-like structures. And so you're going to predict how different is the binding to HIV reverse transcriptase. And this was all done in the absence of a crystal structure for any of these, but we had enough results to feel that our structure was right. And there is some of the predictions. So 9, 10, and 11, we predicted that they would be in that order. 11 would be the most potent. We made the compounds, and our collaborator, Karen Anderson's lab, did the assay. This is a cell assay, but the qualitatively is exactly correct. And the activities there go from 900 nanomolar down to, I can't quite read it, but it is 19 nanomolar. So I think, though, you can see the difficulty if you're a human looking at these structures, trying to figure out which one would be the most potent. But the calculations were exactly right in this and many other cases. So we really had proven the value of doing these FEP calculations. So we had many more results for HIV reverse transcriptase. We're trying to make better so-called non-nucleoside inhibitors because the ones that exist, there are a lot of side effects. Also with HIV, as you're all well aware with viruses, they mutate. With HIV, there are lots of mutants that affect the activity. So we had a 2 nanomolar compound in 2006. Here we are with a 2 nanomolar compound 5 years later. The difference is I'm just showing you the activity toward wild type virus. The compound on the left is not very potent against common mutants. Compound on the right is. So there was a lot of extra work done to make the compounds have a broader mutant spectrum. And then this series here, this is, I think, a nice one. We started out with a docking hit, 5 micromolar, and we evolved that into compounds that were sub-nanomolar. So the most potent is about 50 picomolar. And we also would do things, have done things like take this compound that has a cyanovinyl group, which is precedented in drugs, but you always worry about Michael type reactivity. And by FEP, we're able to figure out what type of heterocycle to replace that with. And we end up with this indolazine that is very potent against wild type in the picomolar range, and also very low nanomolar against a very difficult mutant, K103NY181C. Subsequently, we've also looked at covalent inhibitors. You can see it's suggested there by that acrylamide. This has evolved into compounds that are in preclinical development, such as this compound, number 636. We have crystal structures. We have mouse data that's very favorable compared to known drugs. And we also have even formulations of it that have been put into nanoparticles and implants where we can protect the mice for a month on a single dose of these formulations in the implants. All right, so finally, I'm running out of time here. COVID really took too long. So let me see if I can go back a slide. I'm not quite sure how to reverse going forward. How do I reverse?
The smaller.
The bottom of the, yeah, I'm sorry. Sorry about that. Okay, so COVID, I'm going to have to just zoom through. So first of all, this is the life cycle of COVID. COVID, though it's a RNA virus like HIV, the internal workings of it are quite different. There is no reverse transcriptase. There is a protease, however, and we have gone after the main protease. We've done a lot of work with viruses, so we're set up to deal with the assaying. So the way we started with this is we did docking, and this is now early 2020. We were shut out of the lab from March until June. But we did, I talked to Karen, and I said, well, let's work on this. Try to get a hold of the protein. So she needs some plasmid, which she did, also get an assay running, simple fluorescence type assay on the protease activity. And we would do docking, and I started by docking known drugs. So I have a library of 2,000 known drugs, and I asked the people who have to dock them, did consensus docking. We used 4 different docking, 3 different programs, 4 different protocols, get a consensus because we don't believe the scoring of any one. We bought 17 compounds. I looked at 200 myself, picked 17, 14 of them showed some activity in this assay. That was very exciting. The most potent ones were low micromole. I never expected to get a compound that would be nanomoles, but I was hoping to get a starting point. And then I decided the compound I wanted to work with was actually a pretty weak inhibitor, 150 micromolar, called parampentyl. And I could see that the dock structure to me looked reasonable. This is from Glide. I also could see places where I thought it could be improved. There were deficiencies like missing hydrogen bonds. This carbonyl was over there to pick up a hydrogen bond. There was space here. So by very carefully looking at the structures, I could see things that could be improved. Then I did FEP calculations. So FEP calculations to try to confirm that. And so I won't go into it, but then because of the FEP calculations, the 3 initial compounds that we made, this is now June 2020, we assay and they're 10, 6, and 4 micromolar. Okay. And so we then start from there. It took a couple of months later when we got our first crystal structure, but it confirmed the binding mode that we had predicted. Then we make more FEP calculations. We're looking at this substituent, growing it into a channel. And by FEP, we predicted that, for example, propyl would be good. And sure enough, it's 150 nanomolar. And so we continue this type of analysis, more crystal structures. And I recognize, building with the bomb program, that a phenoxy group should be able to fit in this region that's illustrated. And whenever I have a bare phenyl, you're going to do a chlorine and a methyl scan. That tells me that orthomethyl, orthochlorine should be very good. And sure enough, we make this orthochloro compound. It's 18 nanomolar. At this point, we'd only made about 30 compounds. We've gone from 150 micromolar to 30 nanomolar. So this was published in ACS Central Science. And then we also had to study the activity in cells. So that was all activities just in the protease assay. This requires a BL3 facility with full breathing apparatus because you all know that COVID is airborne. So here's our technician doing this. And then we were able to get the assay results. The best compound we have is number 19. And in the cell assay, it's here, it's 80 nanomolar in the infectious cell assay. Johnson & Johnson took this compound, and in their assay, different cell lines, 50 nanomolar. And that can be compared with the Pfizer compound that's in Paxlovid, which is nirmatrelvir, much more complicated structurally. It has a similar potency in the protease assay, but it's much weaker actually in the cell assay. It's 740 nanomolar with a efflux inhibitor, 4 micromolar without. And it also Paxlovid, you have to add a CYP inhibitor. You don't have to with our compound. So here's the final results. This was just published in PNAS. It's preclinical studies on that compound showing a PanLabs panel that shows no off-target activity of any significance. Here, the bottom line's just a dab of hERG inhibition, but a very clean situation. This is the bioavailability, which is excellent for the hydrochloride salt. Here's activity in mice with our compound. You see good protection of the mice up to 6 days. We also have treated the compound in mice with the Merck inhibitor, molnupiravir, which is not a protease inhibitor. It's an RNA polymerase inhibitor. And then the two together are really good. So as a combination therapy, we're suggesting the use of these two compounds as opposed to Paxlovid. So the long story short, we came from doing butane in Leonard-Jones liquid to developing very quickly. Again, in 2 months' time with a couple of synthetic chemists, we have clinical candidates for treating COVID. So I just want to thank my coworkers, in particular Karen Anderson's group on the assaying, my longtime associate Julian Ferrara-Rose. So thanks for going through this little journey with me here today.
Very much. We have a very, very comprehensive overview of developing methods, which originally were used to understand the today very simple potential energy landscape of the butane up to the discovery in 2 months of a molecule which is more potent than what Pfizer put in the market for treating COVID. Also, there were some keywords which are very interesting. Then I leave the stage for a question and answer. The reaction prediction was AI in 1975. QSPR is statistical learning, and 2000. And the bomb was generative AI.
It's a generative program.
Keywords that nowadays are very fancy and used everywhere for making our papers more fancy for the scientific community. Now the stage is yours for question, for technical question. Remember that then we have 1 hour interview for other kind of questions, but please feel free to ask anything you would like to ask to Bill about this scientific journey. Yes, please.
So it's maybe a naive question, but you said the viruses, of course, mutate, and you want to adapt your drugs so that they can be good against all the mutations. So how would you have to mutate the chemicals? Is that something generally you can say, or does it depend on the specific virus?
So the mutation issue we have studied most with HIV reverse transcriptase, where upon clinical treatment of patients with these non-nucleoside inhibitors, you see mutations arising in the vicinity of the binding site. And so we know about these mutations, and we've been able to make molecules that are positioned better in the binding site to avoid certain mutations. And it's just been, I'd say, a lot of systematic work on the different mutations by doing FEP calculations on mutants to see if we can find something that maintains activity against wild type, but is also still active against the mutant. So there's been some trial and error there, but we've ended up with compounds that have very broad mutant spectrum. With COVID, it hasn't been much of a problem yet. So with the protease, there aren't known mutants, to my knowledge, that are close in the binding site. I think as more people are treated with Paxlovid, one will see more mutants arise. But it helps in general in dealing with mutants to keep your compounds small. Because if they're big, then it's very easy for the virus to figure out how to poke them. So I call there are two types of mutants. There's a poke and there's a shrink. So the poke just makes a steric problem. The shrink takes away a good interaction. It's like Y181C is a shrink. Tyrosine's making nice interaction with a lot of inhibitors. When it goes to cysteine, that's gone. And so we worry about pokes and shrinks.
Other questions? We have also people remotely connected, and we can move then the discussion to the second part. Thank you again, Bill. The second lecture will be by Professor Wodak from Free University of Brussels. Shoshana got her PhD from the Columbia University in New York, and where she started to develop computational tools for biology. Then when she moved back to Europe, she brought these new methodologies in Europe here, and also thanks to the collaboration with CECAM in the 70s, she started to develop methodologies for protein-protein docking and methodology to apply the design of new proteins for certain pathological conditions. Strong collaboration with bioinformaticians, strong collaboration also with clinicians, in particular with the Hospital for Sick Children in Toronto. And together with these groups and these centers, she designed tools for protein networking and protein-protein binding. Between 1886 and 93, she was the scientific director of protein engineering team at Plant Genetic System in Belgium. And she was the co-director of the Center for Structural Biology and Bioinformatics, also leading a master in bioinformatics. And since 90, she is a member of one of the most prestigious scientific organization in Europe, which is the EMBO, the European Molecular Biology Organization. Without any further delay, I leave the stage to Anastasia. Today, Shoshana will share with us a presentation titled Modeling Protein-Protein Interaction, Then and Now. Thank you again very much, Shoshana, for being here, and the stage is yours.
Right. So thank you very much for inviting me for this interesting event, and it's wonderful to be back at CECAM. And you will see CECAM has played an important role in my early career and inspired me a lot over the years. And so it's very nice to see that many people are here. And I'm going to tell you about one aspect of my activities and my interest, which is protein-protein interactions. And I did many other things as well, but this was a continuing line of work that I did until recently. So the outline of my talk, I'm going to highlight the crucial role of protein-protein interactions in the cell. And I will continue by explaining, by presenting what happened in the early days about modeling protein-protein interactions. And then come to the 21st century, modeling protein-protein interactions now, and the challenges ahead. So you certainly all know, but it's nice to remember that proteins play a very crucial role in cellular processes. We have in the human genome, for example, we have 20,000 protein coding genes, and human cells express 200,000 different proteins, not all at the same time. And yes, the proteins carry out a variety of functions. They catalyze chemical reactions. Enzymes, for example, act as transporters and receptors. relay information and regulate one another, and they act as structural building blocks and so on. Now, proteins don't function alone. They function together, they interact functionally, and they interact physically. And these interactions have been selected by evolution to enable them to function in the cellular environment. Now, an important aspect of the research that we are all really concerned with is what is the proteome organization? What is the organization of the proteome in the cell? So proteins and protein complexes are definitely not evenly distributed in the cell, but they are localized to specialized compartments and dynamic membranous organelles that have actually been discovered in the last 15 years and become a very center of research. And these compartments are extremely crowded, very crowded. The concentration of the proteins are very high in these compartments. We have examples, schematic examples of this type of compartments and the concentrations of proteins in there that is very high. And in these environments, specific function interactions, those that have been selected by evolution, often compete with non-specific spurious interactions, which are there and have always been around. And the abundant proteins, if you have high concentration of proteins, the abundant proteins are more likely to engage in spurious interactions. So there's always a competition between spurious interactions and more specific interactions, and everything that happens in the cell is concentration dependent. Now, investigating the protein organization in the cell, the proteome organization in the cell, has been a very, very active area of research, in the 90s and beginning of the 2000s. So to learn how cells function, a first important step is to find out how specific proteins are distributed inside the cell and the interaction they form. So the approaches that have been used, and I have been actually involved in some of this research in Toronto, is to probe these protein interactions using high throughput techniques, for example, to identify binary interactions in the cell, using various indirect methods, and also using affinity purifications to identify complexes. But affinity purifications, both type of approaches give you actually information as information networks of interactions. Protein A interacts with protein B, and the binary networks are very different from the co-complex networks. And we always know that there was a very limited overlap between these two types of networks. And there were also different aspects in terms of what they really identify. For example, the binary interactions miss a lot of protein-protein interactions, whereas the co-complex interactions actually include a number of false positive interactions. In other words, these non-specific interactions that can be detected but are not really functionally important. So if you want to obtain information on complexes from these data, you need to parse these networks into the complexes to get an abstracted view of protein A interacting with protein B directly or indirectly. And all this is averaged over compartments and over cells. And these approaches actually detect mostly stable, high affinity protein-protein interactions. And those are only a fraction of the actual interactions that occur in the cell. So if you really want to know what the stoichiometry of the complex is, if you want to know which proteins interact physically, if you want to know what is their binding affinity, or which residues form the recognition interface, you need biochemistry, biophysics, structural biology, and computational biology. So, this is an example, the network of the proteins that interact, network of interacting proteins for the proteasome. This is actually the structural data that corresponds to this network. So how did it all start? You know, these more, you know, approaches, more computational approaches, more biophysical approaches. So protein modeling, now I'm going to tell you about the early days in which I have been directly involved. I will tell you about the birth of docking, where we docked the bovine trypsin inhibitor interaction with trypsin and the reaction pathway of the quaternary transition in hemoglobin. So in the early days, the influences that the people who played an important role in these early approaches were Cyrus Leventhal, who was my thesis advisor at the time, and who developed one of the very first graphical programs and basically modeling programs for proteins and protein interactions later on. And so this was what happened in New York. This is where things started in New York City in the US. And then, you know, I returned to Brussels, but I interacted closely with Joël Janin, who was at the Pasteur Institute at the time, and who was an enzymologist, but really very, very interested in protein-protein interactions, and with whom he has also had a very interesting biophysical background, and with whom we started to develop, really encouraged me to develop this method. And he also introduced me to CECAM, and this was CECAM at the time was in Paris, and it was headed by Karl Moser. And I'm also citing Levitt, who was part of the important, I mean, I'm citing Karl Moser because he played a really important role, but you know, the very first contact with him was during the 1976 workshop at CECAM that really started, you know, the computation approaches, computer simulation, molecular dynamic simulations, but I did docking at these workshops. So what about docking? Protein-protein docking, as originally defined, was to derive the atomic model of the protein assembly or the protein complex from the three-dimensional structure or coordinates of its components. So this is an example here. You know, you use the two protein component, the receptor, and the ligand, if you want to call it this way, and you sampled all the interaction modes of the ligand onto the receptor. And this applied to assemblies for which the individual component exists in the free form, which obviously is not always the case. And so the challenges of this docking process, or really docking calculations, or predicting doing the docking, predicting the structure of the components, was the structure of the complex, was that you needed efficient sampling methods of rigid body degrees of freedom, as well as alternative conformations, basically the internal degrees of freedom. So you needed the rigid body and the conformation changes. And this was a sampling aspect. But what also, you know, if you sample then many docking poses, and that's similar to what happens for protein ligand docking, you need to identify stable association modes from a very large ensemble of docking solutions. So this requires robust and reliable scoring energy functions to score this position. To do this at the time, there were we had to do this using very inefficient computer systems. And this is an example of what we were using at Columbia, and actually a number of years later, that we needed the graphics, graphics hardware, which was called Adage at the time. It was based on very different technology, was a vector generator technology, not what we have today. And also the Evans and Sutherland graphics. And it was one example was at Cyrus Leventhal laboratory. And we had black and white graphical, pictures. That was the molecular graphics at Columbia University, but it continued a number of years after that. But the graphics was not enough. You needed the computational core. And at the time, the computations were done at Columbia University on another part of the campus by a very large computer, the IBM 36091, and this very, very large computer did what today, your laptop can do. So we had to use it. So we had to use this system, and what we used was the computer graphics program that was developed by Cyrus Leventhal and his colleagues. And this computer graphics program was using the very first versions of force fields for computing the interactions between atoms and residues, and allowed us to do optimization of energy, actually energy minimization, and changing the internal parameters. So when part of my study, when I was still doing my PhD analysis, was to try to use this computer graphics programs, but we had at the time we had a problem. That's where we tried to use it for the first time. We actually solved the structure of RNase S, an enzyme, the structure of this RNase S enzyme in interaction with the dinucleotide, because someone in the lab was really interested in the structures of dinucleotide, but couldn't solve the dinucleotide independently. So we decided it would be, I thought it would be an idea to try to produce a co-crystal structure of RNase with the dinucleotide, and maybe solve the structure of this dinucleotide when bound to the protein. So we used the cytidine 2' 5' adenosine, which was an artificial dinucleotide, which is not cleaved by the enzyme. And we did a difference Fourier where we diffused the small molecule into the crystal, and we got a picture of the interactions. But the apo structure without the small molecule bound was done with sulfate, and sulfate occupied exactly the same position as the phosphate in this dinucleotide, so we couldn't see it. So we thought to really get a picture of what's going on, we needed to do some kind of docking, some adjustment of the conformation of the dinucleotide and of the enzyme, as well as rigid body moving the nucleotide in the active site. But the programs that were available at the time at Columbia were using actually internal degrees of freedom. In other words, rotation around bonds, not actually what the later programs, the molecular simulation programs and dissimulation were using, actually the movements of atomic coordinates. So I needed to do some special, tricks. So to model translations and rotations, rigid body translations and rotations, we needed to use pseudoatoms. So for example, for translation, we needed to put in an axis, a pseudo-axis, very, very long, 100 angstrom from the center of gravity of the small molecule to get translation in different directions. But it worked, and we used it. We changed, the degrees of freedom, the rigid degrees of freedom, and we optimized structure. And this was, I think, seems to be a very first attempt at docking, and it was done in my thesis in 1970. And later on, we kind of applied similar approaches, and then it was while I was leaving Columbia and coming back to Europe. And I was doing docking, together with Joël Janin. And this was also what I really tried to optimize at the CECAM workshop in 1976. So we used to dock two molecules, we used again a polar coordinate system that represented the 6 degrees of freedom. We had kind of two Eulerian angles that positioned. These are the two molecules, molecule A and B, and the center of gravity of the two molecules. And there were two rotation angles, latitude and longitude of molecule one in the framework of molecule one that positioned the center of gravity of molecule two and then rotated molecule 2 using the 2 angles at molecule 2 center. And then you had the center to center distances, and you had a spin angle around the line that was around the vector that was linking the 2 centers. And you needed to optimize, actually, for all these 5 degrees of freedom, were angular degrees of freedom, and then you optimized the interactions in 1D along the center to center distance. But what to do this, it was a very complex program, very, very complex approach. Computers were very basic, not very efficient. So we needed to use a simplified model for these proteins. So, so the simplified model was actually inspired, was actually taken from Levitt at the time, because he just published his simplified model for protein folding, 1 interaction center per residue, and it had a non-bonded term for the interaction, kind of a soft non-bonded term. And then also he added a pairwise solvation term that was also important. He knew that was key, because we knew that it was a key contribution to the binding free energy, but there was no pairwise way of calculating this. So we took his model, which was actually a sigmoidal function, which kind of changed as you approach the residues. So the total energy it was calculated and optimized was like this. So what we applied it to is to the complex of the bovine trypsin inhibitor and trypsin. The structure of this complex just arose not long ago before we did this. And we thought it would be an interesting system to try to see whether we can recall the interactions and how we can, how we can do this. But most interestingly, this really gave us the very first picture of the energy landscape of the interactions between 2 proteins, and that didn't exist before. So we applied the coordinate system, as I tell you, and the potential energy function that was used. 1 interaction, I needed to really make it clear that we used 1 interaction center per residue, and that was also the parameters for this interaction, the size of the balls around each residue were also taken from Levitt. So we actually kind of tested or kind of sampled all the possible interactions of this surface of BBTI into the active site of trypsin. We didn't sample all the trypsin surface, just the active site, around the active site. And what I'm showing here is that this is the energy landscape, and this is the energy, the actual non-bonded and not bonded, the total energy that was computed, and you can see that the best, basically the best total energy was actually the native position, the native pose, and this is shown here on the surface of the BBTI. But what we actually found out is that there are other very kind of favorable interactions, which we call intermediates, non-native interfaces, which are here. Here is a native interface on the completely other side of the protein, and it actually makes sense because the protein was of a kind of oval shape. So it was reasonable that there was some complementarity on the other side of the protein. But this was really the first glimpse of a landscape of protein-protein interaction, energy landscape. But we thought that this approach was needed some improvements, and so we did actually derive a somewhat different energy function. And the most important part of this different energy function was a pairwise, an analytical pairwise approximation to the buried surface area. And at the time, Cyrus Chosia and Fred Richards and Yale, and also B.K. Lee were actually derived the idea of computing the solvent accessible surface area. And Cyrus Josia demonstrated that, actually the buried surface area, the change of surface area upon interaction of the two proteins, of two molecules, doesn't need to be proteins, is actually the solvation energy is proportional to this change. And the proportionality coefficient here, gamma, is 25 calories per 1 angstrom squared barium. But this is a many-body problem, and we derived an analytical approximation, which was could be derived, and was really demonstrated to be actually quite accurate on the residue level, because it really measured. It was derived for this particular model where we have one interaction center per residue. But Clark still later on actually extended it to the atomic level, and it's actually used until today in some of these in some programs for atomic interactions. And so with this new approach, actually what I didn't show and I didn't highlight is that, the non-bonded energy was actually limited to the repulsive part. So the repulsive part was a soft non-bonded energy, while the attractive part was the surface area buried. And we applied this at the time. There was a big interest in the reaction pathway of the quaternary structure change in hemoglobin. So we knew the structures of the hemoglobin in the R and the T states were resolved. And it was a center of interest for the allosteric model of Wyman and Changeux, at the time, Wyman and Changeux. And so we thought it would be interesting to try to model the transition between the R and T states and see how they are related to the difference in the conformations, in the internal degrees of freedom conformations of the R state and the T state. So we performed docking. We used the R tertiary structure or the T tertiary structure for the alpha beta dimer 3-dimensional structures. We imposed a twofold symmetry. We sampled a wide range of dimer-dimer orientations, and we optimized the buried surface area only by rigid body adjustments while minimizing the clashes. And what actually I show here, just a little bit a tip of what we found, is that the transition that we identified, the reaction pathway was not a linear pathway between the R and the T state, but it actually the centers of mass of the alpha beta dimers were actually making a squiggle like this. And this was against what Martin Karplus at the time proposed. And we had a lot of discussions about it in CECAM workshops, which were held in Brussels. And so we imposed symmetry. So instead of 6 degrees of freedom, we had 3 degrees of freedom. We had the theta and phi angles and the chi spin angle. And we started, here is what we found out. We started from the T conformation, and we sampled a wide range of interactions between the two dimers. And we tried to, this is a map that shows you shows you the region of these parameters where we could still obtain a 2000 angstrom squared, buried surface area. So this was what necessary to have a stable complex. But when we started with the R conformation, we couldn't maintain the surface area at that level. And it shows you here that, this is the only contour level at 200 angstrom squared. So actually, we concluded that the transition state between the R and T states were actually more R-like. than T-light, because once, if you want to optimize the energy, the surface area, you had to change conformations. So actually, this was what we published. We couldn't publish this because no one believed us, but we finally managed to publish it in biopolymers. And about 6 years later, Bill Eaton at the NIH and his colleagues, and Jane Hofrichter actually they were hemoglobin experts, and they actually used experimental data on the activation, and they used experimental data, time-resolved absorption spectroscopy to compute the activation free energy for the transition in trout hemoglobin. And they basically used the linear free energy relation to explain, and they basically found the same things as we found, and they actually cited in the abstract. So they said, they basically cite the work that we published, and actually it was very nice to see an experimental verification for this approach. So protein-protein interactions 3 decades later. And this is what I'm going to show you now, not really my work, but I was involved in it because I followed very closely what was happening in the community, and I'll explain to you why. But, definitely, 3 decades later, we had a completely different landscape of data and computational approaches. So much of what we learned about proteins and protein interactions is actually due to progress across fields and across methods. There were a lot of contributions from experimental studies. We had very large number, a growing number of protein interactions, of protein complexes in the PDB, of protein structures, of the structures of individual subunits. And we derived rules, was by machine learning, by what we call today AI approaches. You gained understanding of what is really happening, what matters for protein-protein interactions, what drives the affinity of protein-protein interactions. And we learned a lot about folding, you classified the structures, and you had sequence information, and you linked to in a very interesting and fruitful way. But this helped also to develop new approaches and extend the approaches, extend the simple docking approaches. What I talked to you about is in the picture here. It's basically docking what was done before. You needed the unbound conformation of the proteins to derive the structure of the complex. But later on, as more sequence information was becoming available, more structural information becoming available, you actually were able to do something very different, basically use template-based modeling. In other words, rather than doing the docking, ab initio docking calculations, you looked in the PDB to see whether already related complexes is available. And you basically adjusted, you basically modeled the sequences of your components onto this new complex. And also you derived the different, you extended this information, the docking and scoring approaches, to these other data sources to do what we call today integrative modeling. And one example is the modeling by Charlie, group of Charlie and colleagues of the nuclear core. And also there was quite a bit of activity in ab initio structure prediction of single proteins based on the availability of many structures in the PDB. And also these modeling of single chains really leveraged the availability of structures which were related to the structure you wanted to predict. And hence you could use the known structure as template to build your models. So all this happened, but still no structural information for most proteins is available. For most proteins that have known sequences, no structure information was available for these, for these sequences at all. And even less information was available on three-dimensional structure of complexes because it was more difficult to obtain information crystallographically, especially large assemblies and large complexes. And this information that we did obtain for the multi-structure was really snapshots of single conformations in X-ray and cryo-images. So we also had poor performance for ab initio modeling of single chains and complexes. There was progress, but the performance was still poor, especially if you didn't have template information. If you needed to derive the three-dimensional structure of the complex, of the single subunit or the complex directly from sequence information. And there was also limited performance for other modeling tasks, especially for protein complexes. Now, the status of protein-protein docking in the modeling protein assemblies by the end of 2020, actually, there was a lot of progress that has been achieved to sample rigid body degrees of freedom. And this has become much more efficient thanks to methods based on fast Fourier transforms and on fast Fouriers in polar coordinate method that use that and enhanced Monte Carlo sampling. This was done by different members of the community all over the world. They have really, really produced this. For example, the FFT based method for sampling was introduced by Ilya Baxer together with Kachalski at the Weizmann, and the fast Fourier transform in polar coordinates was introduced by David Ritchie. And this was really has had a very enormous impact on the docking and modeling of complexes. The scoring functions have also significantly improved. And these scoring functions were based increasingly on analysis from examples of known structures. So if you take what Bill Jorgensen was telling us before, they were machine learning based. And these machine learning based approaches were already available at the time. But what was not good or poorly done is handling of protein flexibility and conformational changes. But there was some progress achieved for modeling protein peptide complexes because the flexibility of the peptide wasn't easier to predict when bound to the protein. And of course, there was a very still poor performance for modeling complexes with other macromolecules like DNA, RNA, and mainly due to the fact that this poor modeling of nucleic acid conformations. Now, a very important role in this domain of research was played by the critical assessment of predicted interaction by the blind prediction methods. This was CAPRI, and actually CAPRI is a community-wide double-blind experiment that was modeled after CASP. It was launched in 2001, and its aims were to assess the performance of protein docking and scoring algorithms. And it created a community which was very tightly related, and it was very friendly community. And it dealt with the prediction of protein structures, of the structure of unpublished protein and protein DNA complex, protein peptide complexes, and at some point was also extended to the prediction of binding affinities, but only in some cases, and in interface water molecules. So the CAPRI management team is now headed by Alexandre Bonvin. Joël Janat headed it for a number of years, and I headed it for over 15 years. And a key role was played by CAPRI in fostering the progress for methods of predicting the structure of protein complexes and evaluating the quality of predicted models. And since 2014, we collaborated closely with CASP on the prediction of protein complexes. But in 2020, the CASP 14, there was a resounding breakthrough of AlphaFold 2 from Google DeepMind in predicting the structure of individual subunits, ab initio, and using templates. And it was by one group. The 2 years before that, the DeepMind was doing well as well, but not as well as in 2020, where they have changed completely the prediction, their machine learning model. So this was a resounding change, and it was extremely important. And what I'm not going to explain, this is the AlphaFold model architecture, but just to say that they have leveraged the information from multiple sequence alignments and combined it with residue pairing information based on structural data in a very complex way and a very successful way. And if you want to read again, it's history now, but the Oxford Protein Informatics Group in 2021 said it was a clever, beautiful idea throughout, but nothing really new. And this kind of echoes what Bill Jorgensen just told us. So the DeepMind, it was, clever, idea throughout, but it also is due to DeepMind's superb engineering and enormous resources, the complexity of the approach is such that trial and error played an enormous role. So the important aspect of this was actually the fact that these models provided you confidence scores of the actual data of the structure that you used, and this was a tremendous achievement. And 2 years on, what happened after that in 2022, this AlphaFold 2, this successful model, was actually tricked to predict complexes with encouraging results. So the first one, AlphaFold, was to predict actually individual conformations, and AlphaFold 2 was tricked to predict complexes with encouraging results. Then there was the advent of AlphaFold Multimer, was trained on protein complexes, gave results that were slightly better in some cases, but not in all. And what was actually fantastic is free access that was provided to AlphaFold DB, to AlphaFold software, and to AlphaFold Multimer software, which really changed the field because everyone could have access to it and do their own job. So the open source resources that were derived from there were very, very useful. In the Capri 15, CASP15 Capri assembly, which was in 2022, there was a tremendous, substantial improvement of performance, but not by one group, by many, many groups. And there were lots of different targets, and the performance was much, much better. And this you can see from this plot. This is the score for particular groups, and the higher the score, the more targets were correctly predicted. And the red part of the bar is the high quality structures that could be predicted using all the tools that were provided thanks to AlphaFold 2. And actually, a number of groups did better than the AlphaFold Multimer itself. And this was also due by the fact that they combined docking, kind of classical docking with AlphaFold predictions and with very good scoring functions also derived from known structures using deep learning techniques. So the challenges ahead, actually accurate prediction of protein-protein complexes is not a solved problem. There are lots of progress. The performance is still poor for antibody the complexes of proteins with antibody nanobody complexes, RNA or protein RNA complexes, also complexes with flexible segments and large assemblies and conformation adjustment for large assemblies. And I just list on the other side, on the right-hand side, of a direction for future developments, which are based on getting more data, developing more databases, more data, new deep learning methods, physics-based for modeling conformation ensembles, and extending these methods to handle protein association. So the CASP16 Capri assembly predictions the challenge is really on now and is running as I speak. And also there are a diverse set of protein complexes as prediction targets, and results will be out in December. So where we go from here, we obviously the AI or deep learning models of AlphaFold 2 could not have been possible without the protein data bank and the plethora of data on three-dimensional structure of proteins, the protein genes and the sequences. And for making progress for protein-protein interactions, you need the similar, similar models to obtain models similar accuracy for protein-protein interactions. You would need a large body of data of the different conformation states of proteins and their context dependence, interactions with other macromolecules and small molecules. Well, where will the body of data come from? More data from cryo-EM, and this is an area that is really developing very fast, and from increased integration of experimental and computational methods in close collaboration with data providers like Wikipedia. So thanks go to the people that I already listed before, CECAM, Carl Moser from CECAM, Cyrus Leventhal for my PhD studies and for inspiration, Joël Janin, and Mark Lenzik and Samir Velankar and Nurul Nazardin from the PDB EBI for the CAPI participation. And I also need to thank CECAM for stimulating workshops and support, for EMBO for bringing me back to Europe in 1976. And I actually also need to thank the Swiss National Science Foundation for my postdoctoral fellowship and the FNRS, the Fonds National de Recherche Scientifique in Belgium, for financing the first advanced computer graphic system in Belgium. Thank you very much.
Very much. Fantastic journey. Impressive, your first 1968 docking program for protein-protein docking, far before the Kuhn's paper about docking of small molecules into proteins. And now questions, technical questions, and we have also, as I said, a more informal discussion if you wish, but please feel free to ask questions. Also those remotely connected. I don't know whether there are questions, Sarah.
We have a very shy remote audience today.
Okay.
Shy or small?
No, it's not small.
Shy, yes.
Okay. I mean, ask questions. I mean, I went through, a whole field very quickly, and
And again, we understood that the concept of statistical learning, machine learning was already there far before the keywords that we are using now.
of course. I mean, the success of AlphaFold 2 built on a maturing field and maturing, the data was there, the approaches were there, but you kind of had to put it together. And they had these incredible resources. I mean, these were very, very large, systems, huge systems. Even today, you cannot do any simulations on some of these systems. So you needed somehow, to try to mine the data in a way that would help you to do this. And just the fact that they could predict correctly or high accuracy the independence was giving you already a boost. But when people, and the fact that they made the data, the software, the AlphaFold software was made available, and it was excellent software. People could actually retrain it. So it's an enormous gift to me. I mean, I thought this was the most incredible thing that happened.
So we do have a question from Katrina, but Hindi. But thanks you for these inspiring talks. And then we'd like to know how you can justify that the protein structure obtained from homology modeling or AlphaFold is actually valid.
Yeah, well, actually, there were, as I didn't talk about this, but what was really important is that when the software became available in 2022, already actually helped these predicted structures, helped to solve structures because they could be fit into the electron density either from cryo-EM, cryo-EM studies, or actually X-ray studies, and help to really solve. So you could, the accuracy of these structures was, crystalline. I mean, like the accuracy of X-ray structures, and it could fit into the electron densities it was experimentally determined, and actually helped the structural biologists to obtain actually the refined structure, because in some cases they couldn't identify part of the structure, or they they were stuck, and this helped them. So there was definitely from everywhere, there was definitely signs that in many cases, maybe the majority of the cases not, but in many cases it was reliable information. And the fact that it was attached the confidence scores, and when systems where you have disordered regions in the protein, the confidence scores was often the low confidence regions were often those which are not well structured. I mean, it's ongoing, ongoing evaluation. And of course, in the database, in the AlphaFold database, you have so many structures, you have over 200 million structures. So really, it's an ongoing analysis of whether these structures are real, which part of these structures are real and which not.
Thank you very much. Other questions? Otherwise, we thank Shoshana again. We have now a very quick break to organize the informal interview. So be back in 5 to 10 minutes, and then we start the informal part of our today conversation. Okay, well, welcome back. We have now the second part of our CECAM Marvel Classics, which is the informal part of our conversation, our colloquium. The stage is, of course, yours. I simply start to break the ice, also because I know Bill since many years, and what I have in mind fits also very well on what just Shoshana said during the wonderful presentation and journey. Everyone in my field, which is computational drug discovery, knows pretty well this review published by Bill Jorgens and Science in 2004. And the start of the review is, to me, is very, very nice and it's fantastic. And quoting is, the starting is there really a case where a drug that's on the market was designed by a computation? When asked this, I invoke the professional mantra, every single question is a good question. And this was the start of this review. And of course, this brings to the point of the kind of historical separation between experiments and computation, between experimental people and theoreticians. And my question for you, Bill, and for you, Shoshana, also linked to one of your slides, reporting that very important breakthrough in science can come only through a real multidisciplinary approach, structural biology, biophysics, computational biology. My question is whether your perception is still that there is this kind of separation between experimentalists and theoretical people. Still today, this review was written in 2004, so 20 years ago. Still today, 20 years after all the situation is changing. The contribution of the theoretical community has been remarkable, which I believe has been remarkable over the last 20 years. And so connection and multidisciplinary activities are growing and bringing really to remarkable breakthrough. Also linked to the second question I have in mind, but then at the stage, the others. Please, Bill and Shoshana.
Well, I think that there's been a lot of progress in the last 20 years. And the key word here is collaboration. So drug discovery at this point is a collaborative effort between computational people and, of course, experimental people. experimental people include synthetic chemists who are making molecules. They include medicinal chemists who are sensitive to ADME issues to molecules. They include structural biologists who are often maintaining crystal structures with complexes with candidate molecules with proteins and now nucleic acids. And of course, then with people who are doing the assay work, maybe more on the biology side. So all of these activities come together in any pharmaceutical company. And I think all pharmaceutical companies have very substantial computational groups that are fully integrated into the project. So there isn't, as I said in this original article, there isn't some light bulb moment where somebody goes, that's going to be the drug. They just have to synthesize that molecule, test it. Next thing you know, it's an approved drug. So that just simply, it didn't happen back then, certainly doesn't happen now. So, but the good news is it is collaboration is strong. I think the people involved appreciate each other in a way that, or the level, wasn't true 20 years ago. So there was definitely, I'd say, some hostility for traditional medicinal chemists towards computation. That hostility, I think, at this point has vanished. Everybody recognizes what computation is adding to the other areas of the synergy of modern drug discovery.
Share in the physics-based biology. It has been your experience.
Yes, well, there was always also, the reluctance of structural biologists, experimentalists, to accept the computational predictions of the computational modeling. But in fact, X-ray crystallography, relies on modeling directly, to models. You use information from chemistry, from the properties of conformations of amino acids, to fit into electron density. But this kind of you have computational methods to do it efficiently. So this was the way, they've always been relying on computational methods. But in terms of really modeling, new structures without the crystallographic data, that is something that they always, kind of looked with suspicion. And now, more recently, with AlphaFold structures came out, especially recently, I think the whole field changed. Really, I would say it's a step forward to recognize that it could be useful because they use the predictions to try to improve their refinements. So this was definitely important. That it's bringing together more and more. The fact that you have all these structures in the PDB, these predictive structures in the PDB, and actually the pharmaceutical industry is starting to use these structures to do ligand binding drug protein interactions. So there is a very close, I would say close intertwining of all these actual approaches into it needs to really happen. But I think now the terrain is really right, to move forward in the
That you also link to these message to the audience. Nowadays, the competition is my experience. I think your experience, everyone experience. The competition is not simply between experimentalists and professional people. It's not simply between my group, your group, and the other group. There are the big players. There are elephants in the room, more than one, which is the industrial research activities. They started to play a game that probably 20, 30 years ago they were playing that much like mine without all the Microsoft research and technology, Google research, Amazon research, and which is a new scenario. You mentioned before a very interesting anecdote, Shoshana, about what happened. The failure of our community in predicting protein folding was a very direct question from a very prestigious academy, and then I leave this to Shoshana.
Yeah, it was.
And the competition is this one now, is between also between academic research and industrial research groups, which have a power which is not which is unfair in terms of competition. Which is your point, Shoshana?
Yeah, I mean, I just have to say it's what I experienced during the Nobel Symposium, about a year or two ago, actually after the AlphaFold 2, success story. And it was a big discussion. The people from AlphaFold were there. John Jacobs, he's a brilliant guy. He really is a brilliant guy. I mean, his knowledge of protein structure is great, and also, he understands the AI aspects very well. He's good at statistics and everything. So he was he was very articulate also. He was in the audience. So, what happened there was a professor, really very prestigious professor, kind of asked the audience, how come the modeling community, we are modelers, how come we failed? We tried so many years, to get to the point where we predict really structures with high accuracy. How come we failed? How come they succeeded? So there was a discussion of what happened and so on. And at some point I said, listen, I think it's really the engineering field of these people, the software engineering and the engineering of all these different components that really was very important. And I was moved by the audience. No, there were 60 people or 70 people in the room, and they booed me to death. I was, really impressed. By then, I found this blog on the internet, which actually was written by the Oxford, computational biology group, and they analyzed a very nice blog that explains, the model of AlphaFold 2, and they basically said there is nothing really earth-shaking new. Fantastic ideas. I'm not belittling their contribution, but it could not have happened without the fantastic engineering and without trial and error, because the components that of the different model that they developed for training were so complicated that they had to adjust, ad hoc adjustments of different aspects. That was, couldn't be, it required engineering and required trial and error. So I think that is a lesson. And nowadays, actually, other groups, for example, David Baker's group, the community that he created is fantastic. And, people from different countries were trained in his lab, all over Europe, they are in the States and elsewhere, and they all participate in adding components to their programs. And they have developed similar approaches based on the ideas of AlphaFold 2, but also new ideas, using different models like diffusion models and so on to do prediction of structures and to do protein design. In the area of protein design now is really developing very fast due to this very, I would say, very advanced approaches. And they are AI-based and are really doing very interesting.
Yeah, I think that the keyword is just software engineering is very important.
Yes.
It's very important, really, because the ideas probably are there, and we need to make connections in a very professional way. We put this space in a very
I think there are a couple points I'd like to make here. We're talking about AlphaFold. I actually predicted many years ago, 20, 30 years ago, the protein folding problem, I always said, would be solved by what I call library assumptions. But once there were enough structures in the PDB, we could solve the problem effectively by pattern matching procedure. I've also felt all along that trying to do molecular dynamics for folding was going to fail because of I'm very familiar with force fields and their limitations. So it was no surprise. So I never even attempted. The only thing we ever did was protein unfold. It was eating protein up and letting it unfold to try to get some insight on the pathway for folding the time reversal. It was very clear to me that force fields simply aren't up to starting with an extended chain and then folding to the right structure, plus the search problem involved in the sample structure. So I knew that that was not going to work. And that's why I always felt it would be solved by library summers, which means you have enough information of high quality that you can develop an algorithm to predict the structures. By the same token, I think that the AI concept has taken off very much in science, stimulated by the success of AlphaFold. But people need to come to the realization that in other areas, there's simply not the quality of data to train on that AlphaFold was able to train on protein data bank. So for example, for protein ligand binding, which is critical, we would like to have perfect scoring functions for docking programs, like growing program. So why don't we have that? Because the data for it is a big mess. And so the protein ligand binding data, first of all, IC50s are completely useless because they're a random number. They depend on this concentration of substrate. So you need Ki or Kd data, but for different proteins in different labs, the conditions for these experiments are all different. So what's happened though is the docking scoring functions have been trained on a mishmash of data, including IC50s, EC50s from cells, KIs, KDs, and you don't have a large, consistent database to do the training like you have with the PDB. We don't even have it for something like aqueous solubility. You find out in that case, again, different labs are all measuring aqueous solubility in different ways. They're using buffers. The buffers change. So I think that the enthusiasm about AI in general is going to solve the drug discovery problem is way off base, because we just simply don't have the data sets to train on that you need to have the type of success you've seen through the PDB yield and
Okay, so I mean, there is a technical question here for Gil, but in the spirit of the general discussion, because there are a couple of things that resonated from your conversation now that I wonder, are they transferable to other simulation communities? For example, the material simulation community, because this insistence on the relevance of having high quality data train on. I don't even know how the PDB was born, and it's amazing that such a large community was able to somehow converge on a unique infrastructure. So,
can I say something about how it was born in 1972?
I would love to.
There were a few people that signed a petition to the NIH to form, the time it took to really form the PDB. It was signed by Helen Berman, Joe Sussman, myself, and some other people. And this is how it started with a few, four or five structures, and maybe 7 structures in total. And this is how it all started. And the vision was that it was absolutely necessary to have this database. Otherwise, the vision of the importance of the data was already.
Yeah, and the community followed.
Was mandatory.
Slowly, I mean, it was a battle, because, the publications, the journal had to insist that this structure, before it was published, and that took a number of years. So it was a long, a long journey. But, only, until 20 years ago, it was too big. And now today, with some infrastructures, some structures are very competitive, though they ask not to release them for 6 months or a year, something like that, so you have not access to them. But it's getting better in order. Now that people realize how important it is to data, I think it becomes easy. And also, we need structures, to use as targets for our blind predictions, and that is also a battle. Recently it has become easier, and I think the structural biologists, really realize this. But I wanted just, to make one remark that actually so the prediction of the native structure. But the process, the mechanism of folding, they didn't do anything about this because they actually produce single structures. There is information there, you can extract information in multiple structures, and maybe some intermediates. And actually, when you look with how these structures, how the model increase the structures, there is some videos that shows you how kind of locally, you have local interaction on local structures and the local structure can propagate. This is something that we learned in protein folding, the importance of local interaction, side chain, nature interaction, local formation of super-secondary structure and secondary structures. So yeah, I mean there may be something there about the folding that is not extracted.
Yes, the protein folding has not been solved. We have a way to do the structure of this. And on top of what Shoshana just said, now 90% of journals is mandatory to deposit the structure in the PDB with the quality control, and without the deposition of the structure, you can publish your paper. So now it's even more stringent than a community-driven effort. So either you deposit your structure in the protein data bank or you don't publish your paper. And after the deposit, there is a quality control of the data. So now the data in the protein data bank are of super high quality, and this is why they are very reliable.
It's gotten better, but it's gotten much better because now you also need to deposit the structure factor.
Yeah.
And for many years, the quality of protein structures was evaluated based on the programs that analyze the structures that were already deposited in proteins. And that is, these were the early programs that were developed by Janet Thornton. But this is not enough because you didn't know how actually these works fit into density. So, in analysis, quality control, that actually, we build the structures to see how it fits into the density. Now you need to submit also structure.
So, there are two aspects to the protein folding problem. To many people, the protein folding problem is, can you predict the three-dimensional structure of a protein given its sequence? And that is what AlphaFold does. And so that is a goal. The actual pathway, if we would add the unfolded protein in solution, that is a different story. That is the pathway. And keep in mind, many proteins don't fold starting from just being an unfolded structure. One, produced by the ribosome, folding back, etc. So I can't remember what percentage, but I think it's only less than half of proteins, if you follow the nature, refold into state.
Questions from the audience? Yes, please.
So in recent years, we have developed this machine learning for force fields. which are of higher accuracy to the previous ones. So do you think there's still some chance that we should to arrive at the voted for them from the molecular numbers, or do you think it's out of reach?
So there are force fields being developed using machine learning. Again, it's a question, what are they being trained on? And so I haven't, and this is also, of course, appearing in the quantum industry, where people are trying to predict energies of molecules by doing machine training on, for example, cluster results for hundreds of thousands of molecules. So I think there's certainly going to be progress in those areas. The force field problem, you can also train on the same type of data with a couple of cluster calculations on complexes. And so I think that will advance. I haven't seen it making major inroads at this point in comparison with traditional force fields, listen style force fields, which incorporates OPLS and the charm. Those are still definitely the dominant type of force fields. The polarizable force fields have not become a major issue after 30 years of talking about it. There is no generally accepted polarizable force field. So, but we certainly hope that there will be continued advances. Many of the non-polarizable force fields have added special terms for things like halogen bond and cation pi interactions. These are elements that are very much affected by these electronic structure issues that are captured, hopefully, with polarizable force field, but you can add them in a one-on manner to current force fields, and that has made significant improvements. For example, with OPLS-4 has addressed some of these issues. So I think we'll see increased activity in the machine learning force field area and also with quantum chemistry. Probably not a great uptick in activity in polarizable force fields. That'll still stay as a sort of low-level activity. I think at this point, the Drudy force field has some nice results, and I always liked that treatment polarization. It's not perfect.
Yeah, but I would say, you know, for getting good force fields for protein interactions, this is also, you know, missing data, not missing data, not structures of complexes, and actually, you know, the actual affinity measurement, to experimental affinity measurements. And there are several community initiatives that, you know, try to address this, saying we have benchmark data sets for protein-protein interactions, you know, they're being used to develop, you know, force fields, to develop docking methods and so on. But, you know, the force fields that are used to evaluate, you know, this score, binding mode, don't reproduce the free energies, correct? But, you know, the free energies are also measured by using, you know, different systems using different approaches. We wanted at some point, you know, everything done, you know, instruct by experimentalists, you know, to really measure the affinities for a set of example structures and different types of components to see whether it's doable to do it in a completely controlled way. Because really, I mean, all these force fields don't reproduce any experimental data.
Other questions? Yes, please.
I was wondering, there's a lot of methodologies also being present in the past. So how much would you attribute the current success of the AI to an improvement in the methodology versus just having more and and better quality data.
The improvement in the methodology itself that is used to train, to train.
Yeah, like just like I don't know diffusion algorithms, what they are.
The diffusion is being adopted more and more, recently, but especially for protein design. So, they're using, actually, AlphaFold 3 has declared that they're incorporating some diffusion models to really improve. They changed the model for the training model completely, which they say can now add protein, acid and protein small molecules, and that, there was a component, the component of this model that was based on diffusion. So, yes, I mean, the diffusion models have been introduced by a number of people, including others too. So it's not, not surprising that they have some advantages. That also, the heavy language models were introduced at some point. But they're saying that the floating language models, really you can predict the structure just using one sequence. You don't need multiple sequence alignments, but this is a fallacy because the model actually incorporates all sequence data in the model itself. So you don't need to provide the insight, but they use the information.
Again, I come back to what makes the PDB different. So to train scoring functions for protein or protein-related binding, you need to have high quality data that's been obtained in a consistent manner, and that doesn't exist. The PDB is very special because you have hundreds of research groups who have contributed structures that are obtained in a pretty much consistent manner. I mean, there are differences, for example, the pH in which crystals are grown, but that's reported, and one can think about what are the consequences. Predominantly, there's consequences in the protonation of histidines. In extremes, maybe protonation of carboxylic acid. But it's very narrow range of variability, I would say, in the PDB in the way it's structured. You have this huge community all contributing to it. And that just doesn't exist in any other area that I can easily think of. So we can create it. We're talking about this machine learning of training on high quality quantum mechanics. So there we're using computation to generate the data, to do the training. And that certainly can be successful. But if you want to train against experimental data, like PDB, it's going to be tough to get people excited to create consortia to collect data on Ki, Kb data, and protein ligand binding, hundreds of different groups. It's going to be viewed as too mundane. So will be a problem.
So it's a combination of the two. Yeah. The new methodologies over the last 20 years, mainly thanks to the neural network, because since many years are possible to go deeper and deeper. Is one advantage, and second is the data. It's simply the PDB is there, the possible binding is not the data with pHs.
And the computational resources available as well.
And the computational resources, yeah.
This is really computationally intensive stuff.
And then we are back to the software engineering, which are a key part of all the activities. And today, with Sarah, going back to the very, very old archive, this is in the DNA of CECAM, not because of the Sarah. One of the directors applied historically to the second director position in the submission letter for the director said, we need to increase the interaction with the computer center because we need to implement our idea into good software 40 years ago. So is something that we should keep in mind that as a software engineer, the good that she got from the Nobel Symposium was absurd, because they succeed because they have many software engineers available and brilliant people to bring together points and to make something very robust from the statistical and from the computation standpoint.
But wanted to say something because, you know, the protein, you know, protein data, bank data, will explode enough, yes. It's that in the PDB, you have many protein structures that are the same protein is in different environments and interacting with different small molecules with different proteins. And, you know, it's not necessarily, it's like, you know, sampling. You can consider this as samples of the conformation states that protein can adopt alone, because, you know, the actual conditions that are being caused is the conditions that stabilize a certain conformation, which was already sampled by the protein. I mean, the crystal structure of the protein is not necessarily the biggest energy. It's, you know, it's close probably, but so now this is the idea that we started, is to really derive information in samples from protein structures. And I think this could be used to generate samples that can be then used for training, you know, AI approaches and combining the simulations and physics-based approaches to really find out whether these you know, multiple conformations in the PDB, do they you know, can be derived from them, if we cluster them, etc., can we derive conformation states that are really functioning? And I think this is a new area that it really starts happening because of the number of really similar, you know, same proteins, is very similar proteins in different environments. This is very important.
There is an initiative, there is a European project for DDB to collect the dynamic data.
This is different. What we, you know, what I'm suggesting is to use the actual crystal structures, not use NDB. Use the actual crystal structures of proteins in different environments in the PDB. And we basically take that as samples. And then, you know, you don't know, these are experimental ensembles, native proteins that, you know, are a proxy for what the protein will go really sample by itself. And then you need to use, you know, actually computational methods to apply to see what they correspond to. You know, if you cluster these ensembles, you find slightly different states. And do these states really represent the state, or this is, you know, what is it? So the these simulations and other, you know, type of simulations to find out the hinges, to find out what's going on. So it's a way of mining the data from the crystal structures without the inputs as a basis.
Yeah, I have a question that was prompted, I think, by one of the slides of Professor Voda, where the sort of powerful Columbia computational facilities were shown. And then it was also mentioned the first high-powered graphic computer in Belgium. So it's more about how was it to be a computational scientist in the 70s? I guess you wouldn't spend your time in front of a cell phone or of a display. And so what was the practice actually of doing the computational science?
It was actually, you know, magical, because it was the first time that at least I could look, you know, and other people could look at it actually the proteins on the screen and try to interact with this. And it was, you know, really a vision that kind of drove me at least to continue to explore this area, because I thought that this was the future. We needed, you know, physics and understanding of what is really going on and quantify what is going on, you know, to understand what is happening. But I was very isolated, and other colleagues of mine was thinking the same thing. I was doing that. I said, no, I'm not doing that. I'm trying to understand how proteins work. Your proteins work. So I was actually, as a woman, I was quite alone doing this, and not many women. But I was fascinated by the vision, by the possibility of doing this. Then later on, by building bigger, bigger multi-protein structures. What we did, Sarah Capital Lab, is building structure of the fiber for sickle anemia from underlying functions. And actually, our model was wrong, but the model of Perutz was wrong as well. So it doesn't mean that. But it was an experience because the computers were very slow, to standards of today. Then we needed to wait for the computer output and computer output. The outputs were using the job controlling, which is IBM, computers. When I came back to Belgium, we didn't have access to screens to change. We had to use cards. So it was an enterprise. We have to have strong muscles to pull up all the comments. That was given at the first CECAM workshop. Everybody comes.
What about you? Was it similar?
The first computing I did was I was in high school. So this is around 1968. It was a teletype that was connected to a computer in darkness. And you could write little programs in BASIC. And then if you wanted to store the program, it was what's called a paper tape. And you could print the program on the paper tape and you could read it back. The first time I studied computer graphics was when I was at Harvard and working with Corey. And Corey really is the first co-worker on this from Todd Whitke. Whitke developed the first graphics for drawing organic structures that I think ever happened. And this was on a PDP-1 computer at Harvard, in the CECAM laboratory, with Evans and Sutherland system. These were big, round CRTs with a tablet, and you could draw on the tablet and create the organic structure. The PDP-1, so I did a little bit of work on the PDP-1. There were no compilers. Everything was in assembly language. So you had to do that. And then subsequently they got, and by the way, this is Bill Gates was in that computer center, and I had some overlap with his time there. So he was at Harvard, and that's working in the same big room. There was no remote access to anything. You had to be there with the computers. So then after the PDP-1, they got this wonderful thing, the PDP-10. The PDP-10 had a port drinker. So that changed everything. And also at that time, Tektronix came into the scene. There were Tektronix graphics terminals where, again, you could draw on it, but then there was a phosphor. So to change the picture, it had to be erased and redrawn. And then they also had a tablet. And so that was my first experiences. And then Evans and Sutherland stood out with the protein structure display. Very much helped by the digital equipment company, again, coming up with VAX computers. So the PDP-10, then there was a VAX 11780, and they would have attached to them these Evans and Sutherland systems. So the VAX in so almost 1980, was about $250,000. The Evans and Sutherland system was another $150,000. So a setup like that's $400,000 in 1980. You could buy a pretty nice car for $4,000 in 1980. So $400,000. If you put it in today's terms, it's a lot of money. So individual research groups typically couldn't afford those systems. You'd see them in industry. And then slowly, as workstation, as mini computers became more available, and then the type of graphics needed for proteins eventually was done on less expensive systems, but that wasn't until well into the 1980s before there was significant computer graphics. I very much remember visiting Sandoz once, and they had an Evans and Sullivan system. It was the first time I ever saw a CPK structure in color. So now we can rotate CPK structures. But the first time I remember, I was saying, here it comes. We're going to just display the organic molecule with a CPK structure, so with spheres. So here it comes up on the Evans and Sullivan system. It starts at the top. Just making note, because it's computing as fast as it can, is attached to a vaccine. But then after a few minutes, there comes up this picture. You know, the first time I see this, just absolutely wonderful. I remember it distinctly. I was visiting Sandoz, and they decided to show me this thing. It couldn't rotate. Real-time rotation came much later. And Shoshana, I'm sure will remember on the Evans and Sullivan system, Connolly service. So this was a way of displaying protein structures at that time on Evans and Sullivan, where we just used a lot of little dots to make a so-called Connolly surface. Used by a fellow, I think he was in San Francisco, he had worked with Bob Langridge, who was a pioneer in protein graphics. So they're very, very interesting to watch all this evolve. The situation today is wonderful. With your laptop, you can show very complex structures rotating in real time.
I have to say something about, there was nothing in Europe. I mean, they were really lucky to be in the States. When I left the States, come back to Europe and try to continue my work, there was nothing there. So, I started to try to get allies in Belgium to help, to get interested in molecular graphics, and it was quite expensive. So we finally found some allies from 2 different universities to apply for a proposal to the National Science Foundation to buy the system, which consisted of an Evans and Sutherland and the PDB-11 at the time. So we have submitted the project, we got, 7 months later, success. The project was awarded, but when we looked at what was awarded, they awarded only the Evans and Sutherland and not the PDB. What do I do with this? So we decided to find, somewhere on campus and elsewhere in the PDB-11 that we could book the Evans and Sutherland. So we found it at the hospital, one of the three university hospitals that was splitting electrocardiograms. And we attached our computer graphics to the PDB-11, and we had 2 days and 2 nights, something like that, to develop programs. We did that for a number of years, and then we applied for the PDB-11 again, so it was so absurd, to be in this situation. And this is how it all started. I think it was the first molecular graphics in Europe, after the Max Planck Institute with Robert Huber and Vigenos. So we were, like 2 machines in continental Europe, and then we all developed later. We had, the VAX computers and the mini computers, mini high performance computers, etc.
I think we can continue our formal discussion upstairs during the apero. Thank you very much again, Bill and Shoshana, for coming for this very interesting afternoon. And wonderful discussion.